Good afternoon. I'm Gula Lipschitz, and it's now my pleasure to introduce Dr. Greg Lasati, Chief Technology and Manufacturing Officer at Century Therapeutics. Again, the format for this session is a roughly 25-minute fireside chat. If you have questions during the session, feel free to type them into the question box, and we'll try to ask them on your behalf. Greg, thanks for joining us. Can you start by giving us a brief overview of Century and then walking us through your manufacturing process and its advantages?
Thanks, Gula. Thanks for the invitation today. We're happy to be here. I'll tell you just a little bit about Century, but I'm going to spend the majority of the time talking about our manufacturing approach and how we're using scale up and the iPSC platform to reduce cost of goods to the lowest possible level. So Century is a developer of iPSC-derived immune effector cells, both T and NK cells. And we're very excited to be one of very few companies in the clinic with iPSC-derived immune effector cells. We have an NK product in the clinic that has, we believe, the most edits of any product in the clinic. There's 6 edits designed with a CAR, as well as what we call alloevasion edits to evade the immune system to allow us to do multiple dosing. And we believe this is going to help us in the field of lymphoma, where we have the ELIPSE-1 trial currently ongoing, and also in the field of autoimmunity, where we have a trial in lupus about to start, which an IND is cleared for. And we just made an announcement that we have an intention to take that product into other autoimmune indications with additional regulatory filings. So we're very excited to be very, very front and center in this field, and we're excited with our platform of these iPSC-derived immune effector cells. So I think with that, I'll transition to the manufacturing approach, which is one of our great advantages of this platform. Okay, so I just want to start by reminding people of the advantage of allogeneic over autologous, and this applies to both donor-derived as well as iPSC-derived allogeneic in terms of the advantages listed on this slide. And very simply, with autologous, you cannot scale up, you can only scale out, because every patient will require his or her own batch to produce material for their therapy. So there's no way to scale up. You can only do multiple batches in parallel, which doesn't really do too much for you in terms of economies of scale. Whereas with scale up, you can make much larger batches and make many doses that can serve many patients with allo. This is going to give you greater efficiencies. It's going to spread your testing costs, which is quite expensive, over multiple doses. It also spreads your labor and materials, reagents, supplies, et cetera, over multiple doses. This lowers your cost of goods. And also labor obviously is a contributor to cost of goods, but labor in itself can be very challenging, especially for autologous, where the patient numbers are quite large, and hiring just that many people in manufacturing and quality can be quite a challenge to get that many people online and trained. So this gives us a greater opportunity for better capacity using allogeneic, which ultimately will lead to better patient access. I want to make a further distinction about iPSC-derived versus donor-derived allogeneic as well. So in the top bar here, I'm going to try to use a pointer. Let's see if this works. Can you see my pointer okay? Okay, great. So in the top schematic here, this applies to both autologous and to donor-derived allogeneic, in the sense that you're always starting with one person, either the patient in the case of autologous or a healthy donor in the case of allogeneic. You're doing your gene edits, you're making your drug substance, and the difference with donor-derived allogeneic versus autologous is that those products can treat many patients. But you must keep going back to donors to make more product, because there's a finite proliferation potential of the donor cells, and you don't want to exhaust your T cells, for example. So this is going to be limited by the number of doses you can get from one donor. So you have to do your gene editing multiple times. Every time you get a new donor, you have to look for donor-to-donor comparability. The difference with iPSC-derived is we start with the healthy donor. We reprogram their cells to make induced pluripotent stem cells, create a bank of that. Then we do a genetic editing to put on the features we want and to knock out the genes we want to knock out, and ultimately pick a clone that has all of the desired features. From that clone, we can create a master cell bank, and the master cell bank then is the beginning of manufacturing. We never have to go back and get more donors. This genetic modification is done only once. The master cell bank is not only large enough, but also has the proliferative potential to make enough doses for the lifetime of the product. So this is the major distinction, and these are the major distinctions. Again, one donor for the lifetime of the product. You don't have to redo your gene editing. You have a clonal population, which reduces variability because you're going to start with the same starting material every time. And you have no worry about donor-to-donor comparability. And just to summarize a bit here, autologous will always be challenged with the one lot per one patient paradigm. Therefore, the cost of goods will always be the highest possible in terms of these cell therapies. And you really don't have many economies of scale other than perhaps batching some of your tests to reduce a little bit of cost, but you still have to test every dose. Whereas with allogeneic, you're going to spread your doses, spread your costs over many doses. And as I mentioned before, donor-derived will be somewhat limited in terms of scale because it will be capped by the largest number of doses you can make from a single donor. Whereas with iPSC-derived, there really is no limit to scale other than the engineering and biological limits, which I'll talk about in a minute. So with that framing, let's just talk about scale up and what has to be achieved. People think about scale up, they think about growing large numbers of cells, but I really like to compartmentalize this into three major categories. Of course, you have your cell expansion. We grow very large numbers of our iPSCs before we even differentiate them. Again, that's one of our advantages. We're not worried about exhausting our cells because we can grow large numbers of the iPSCs first, and the edited iPSCs in particular. But there's also the harvesting step, to harvest large numbers of cells and to exchange the spent media for excipients that you're going to formulate in. Takes quite a large volume to deal with, and this is not something you're going to do at a lab scale in terms of centrifuges, but you can use things here like a counterflow continuous centrifuge. You could also use tangential flow filtration. And then there's filling. If you're going to fill hopefully thousands of vials, you're going to need to somehow automate that filling to speed it up. You don't want your cells sitting in cryoprotectant very long, and you also don't want them sitting in unhappy conditions, no longer growing at 37 degrees very long. So you need this to be fast, and you also need to inspect every vial per USP1 guidelines that says you must visually inspect for extraneous particulates. So automating these things can be very important. But in general, I just want to say that we are taking a fundamentally engineered approach when looking at all of these things. We have been able to draw upon experience from the biologics and vaccine worlds for a lot of these unit operations. And when you do this, you think about the key scale-up parameters. You put models together to understand the effect of the different input parameters on product outputs, and then ultimately you have to have some scale-up data to refine your models and then adjust them and then further scale up. And I just want to hone in a little bit more on the bioreactor piece. This slide shows some of the bioreactor choices one might have. In terms of scale out, I mentioned that earlier, that's something that autologous is stuck with, but with allogeneic, you can scale up. So here I'm just depicting a adherent type culture transitioning to suspension, but with suspension, you have many choices. You have rocking bags, you have vertical wheels, and then you have traditional stirred tanks with impellers. But in all of these, it's not so straightforward that you can just make larger batches. You have to think about your engineering challenges, which include getting enough oxygen to the cells. Probably more importantly, or more difficult sometimes, is getting enough CO2 removal so the CO2 levels don't become toxic to your cells. Making sure you can mix properly without damaging your cells, and then controlling all your key parameters like pH. And I highlight pH only because it's a tricky balance between the CO2 that you're putting in to keep the pH down with the sodium bicarb buffers you use to help bring the pH up as needed. You don't want to mess up things like osmolality when you're adding sodium bicarb. So it just becomes a major engineering challenge. But again, we can draw, and we have drawn from the field of biologics to use that experience to better design these processes and scale them up. And then if we think about the benefits of scale up, there are quite a few. So the first two rows talk about some of the things you can do to better control your process by, again, back to the gassing strategy to get oxygen in and CO2 out. There's very straightforward engineering principles, which I won't get into here, something called KLA, but that's a subject of another talk. Talk about how we can control this properly. Nutrient levels are much better controlled in these types of systems other than static, where it's basically like a fed batch or a batch system. These can be perfused, so you can constantly be pulling off spent media and putting on fresh, but you do it in a way that's got a control loop that you understand key nutrient levels, and you feed and pull off spent media at the right times in the right way. Of course, we are not spreading our risk among multiple vessels, which then leads to multiple chances of contamination. We have less aseptic manipulation, so that reduces the chance of contamination. But ultimately, these types of vessels and these types of systems could lead to much higher productivity and less use of media, which drives your costs down. They take up less space, less labor intensive than autologous, where every patient is being tended to by really an individual operator. Here, operators can handle these batches, which could supply doses for many patients. And then the other big one is the analytical tests, which are very expensive, but they're now divided by the number of doses in a scalable batch as opposed to autologous, where it's all for one patient. And as you scale up, most of these costs go down linearly. Want to spend a few minutes talking about Century's investment in manufacturing. We've done this a number of ways. We built our own manufacturing facility in Branchburg, New Jersey. This is a 53,000 square foot building. It was designed to be versatile to handle multiple types of immune cell types. And our thought is that having our own facility allows us to learn and address things and pivot more quickly, and therefore iterate faster on products. We're also very privileged to have a great relationship with Fujifilm Cellular Dynamics, who has GMP manufacturing, and we have the ability to make product at either our site or their site. Matter of fact, Century 101, which is in the clinic, has been produced at their facility in Madison, Wisconsin. This gives us great flexibility. And just in general, we believe that our approach to manufacturing and process and analytical development leads to better process and product consistency. Having these scalable platforms, as I mentioned before, we can reduce COGS and meet the demands. Our process, which does not involve expanding final differentiated cells, improves cell fitness. We're not worried about cell exhaustion as you would be with donor-derived. And because we have a clone as our starting material, as I mentioned earlier, we believe the homogeneity of the final product is much greater, and therefore characterization becomes more straightforward. Just a couple of quick pictures and I'll wrap up. This is our facility in Branchburg, New Jersey. Just a few shots of it. It's a really nice building, and it's got very high ceilings, as you can see in these two pictures on the right. Kind of see it here in the main picture as well. But this allowed us to build the GMP facility in a way that was very efficient. It allows maintenance to be very straightforward. And we have a nice process flow here. We have docks on both sides of the building, so raw materials come in one side and finished product goes out the other, which is nice for a GMP flow. And then just a couple of quick pictures. These are the entry and return quarters for the majority of our suites. We do have multiple suites of both grade B and grade C delineation. And then the last slide, I'll just show you a sense of what a suite looks like. This is one of the grade C processing suites during some of our practice runs or demo runs, and a GMP run on the left there. And you can see quite large suites, can handle a lot of operators. We've, of course, done the environmental monitoring performance qualification to prove that we can handle these numbers of people if we need to have a very intense processing day, and just gives you a flavor of the facility. So with that, I'll wrap up, and I think happy to take some questions from Gula and talk about some other issues related to this.
Great. So maybe let's follow up on some of the things that you touched on here. So regarding COGS, it obviously makes sense conceptually that being able to scale up rather than out and use a single clone for the whole product can drive cost reductions. But maybe to expand on that, what are typically the major cost drivers? You touched on this a little bit, but what are the major cost drivers of cell therapies? And how low can your workflow go in terms of reducing COGS?
Right. Yeah. So I think there's a lot of contributors to cost of goods ultimately, but I guess three of the biggest ones would be some of the reagents, and cytokines can get very expensive. The supplies, some of the plastic you use, whether it be in a small device like a Zori Wave, a G-Rex, or even a larger stir tanks, those disposable devices get pretty expensive. And then the testing, we have to do quite a bit of testing on these things to ensure the quality of our product. And some of those are done in-house. We do a lot of flow in-house, for example, but there's also all the safety testing, which gets quite expensive. So being able to spread that out over multiple doses, all those things, the supplies, the reagents, and the testing is very important. And then the labor is a big piece, particularly for autologous. I think it's less so for both donor-derived and iPSC-derived, because we are spreading that labor out among many batches, and many doses. So a single batch can be handled by a couple of operators, and that does hopefully hundreds, thousands of doses perhaps eventually. But with autologous, those couple of operators are handling one patient. So that's where labor becomes less of a contributor to COGS in allogeneic. And then, as I mentioned, with the difference between donor-derived and iPSC-derived, you have this ability to make an unlimited number of doses in a batch. So we believe iPSC-derived will enable the greatest or the lowest possible cost of goods, because we can go to the highest possible scale.
In terms of what aspirations there could look like, are we talking about trying to get into the realm of biologics closer than traditional cell therapies?
Absolutely. Yeah. I mean, I'm not ready to give a number today. There's still too many variables and too many assumptions, but I think based on what we know, as we talked about earlier, the size of these bioreactors, we know the biologics world has done bioreactors up to 20,000 liters. We know vaccines have done bioreactors up to 2,000 liters. And we think based on what we're learning about how our cells handle in these kinds of systems, and we have done work with more than one type of scalable system, we believe that our cost of goods will be more on the closer to the order of biologics than they would be of current autologous cell therapies.
So on the topic of scale, what is the current scale of Century's process? And with the expanding efforts in the autoimmune disease that you mentioned at the beginning of your presentation, do you envision needing to scale up further than perhaps what you built out initially to meet that demand?
Yeah, I'll start with the second question and come back to the first. Yeah, I think autoimmune, we're hoping, I think the whole world is expecting autoimmune to have a greater demand as more patients and more potential indications. And we think that could increase the demand quite a bit. And that's why we're so well positioned, because again, having the most scalable platform means this could be the best approach for those autoimmune indications. Of course, we're very bullish on hematology oncology as well. But when you think about the cost of goods, it might have a much bigger be a bigger factor in autoimmune. And I'm no pricing expert and reimbursement expert, but I would also think the prices will probably need to be lower in autoimmune than they are in oncology, hematology. So again, here's another major advantage of the iPSC platform. So to your first question, we've taken a very measured approach. We've tried not to overinvest in our phase 1 process, and in terms of scale and in terms of the amount of time we spend getting ready for phase 1. It's important to get to the clinic quickly, understand what your product does, but at the same time, and anybody who's heard me talk about manufacturing in the many years I've been in this field, and I've been in cell therapy for almost 20 years now, it's always with this idea that you have a line of sight to commercialization. So you never want to just do something in phase 1 because it's convenient or quick. You want to have a plan and know that I know I can change this process to something scalable later. We're not afraid of process changes, because the key with process changes is you have to understand your product very well and have a whole arsenal of characterization tools, analytical tools to understand your process, and then understand the engineering related to your unit operations, understand the biology in terms of how your cells behave. So again, we've done a very simple approach for phase 1, phase appropriate, not a very large scale, but we have very definitive plans on how we're going to change the platform, scale it up, use these scalable unit operations, some of which I highlighted in the slides. And we do think the autoimmune is going to demand even a greater scale than we first anticipated when we started this business, when hematology oncology was probably just our sole focus at the time. So it's an exciting opportunity for my team for sure, and I think we're really well positioned to handle it.
To what extent do your current processes, or the ones that you might incorporate later on, use things like automation to reduce variability and or increase efficiency?
Yeah, everybody talks about automation with autologous, thinking, okay, we got to remove those operators and save dollars that way. And that's helpful for them, but automation does another thing for you as well. It helps you have process control. And because we're leveraging a lot of technology from the traditional biologics and vaccine worlds, those bioreactors, those harvest systems, the filling, all of that comes with automation built in. So feedback control. So if you understand the effect of your input parameters on your product attributes, which we spent a lot of time doing, I didn't really talk about that so much today. We're always interrogating our processes, trying to understand which parameters affect the intermediate process outcomes, as well as, of course, the final product outcome. And as you understand that, then you can design your processes to have the proper feedback control. I talked about, for example, controlling nutrient levels in your bioreactors. Well, you need to understand what to control them at, but there are certainly these unit operations I talked about, these bioreactors, have all these built-in feedback controls where you have simple probes like pH, even CO2 probes, and you can measure metabolites in line, and then have feedback control then to control those parameters appropriately, so that at the end of the process you have the right kind of product. So yeah, we are constantly taking an engineered approach of having the right process design, so that we understand ultimately then how to control the process to get the final product attributes.
So as you mentioned, your most advanced process is or program, I should say, is in NK cells, but you do also have other types of cells in your pipeline. So how much overlap is there between the process components or equipment there?
Yeah, we've been thinking about that from day one, and we're very fortunate to be able to, again, have that line of sight to commercialization from the very early. So we're always thinking about biologically, how do we make a T cell? Biologically, how do we make an NK cell? But from a scale-up perspective, how can we do that in a scalable system? So we've been trying to have as much synergy and overlap, and we have quite a bit of overlap in terms of the types of bioreactors. We've shown that both our T cells and our NK cells can grow in these dynamic bioreactor conditions. And then when it comes to the harvesting and filling, that's a little more straightforward because cells are cells. Once you try to separate cells from media or fill vials aseptically, that's pretty straightforward. But the bioreactor is the big one, and we're very confident that we're going to be able to streamline things, which will help us in terms of learnings and optimization. So yeah, and we feel pretty good about that. If I get one more thing, if I get one more thing, maybe you're going to ask me this anyway, but
yeah, go ahead.
We just had an acquisition of Clade Therapeutics.
I was going to ask about that.
Okay. Yeah. So we're very fortunate they also thought ahead when it comes to scale up, and they've proven that some of their differentiation or the key differentiation technologies can work, or not only can work, but they've been designed in a way to be amenable to dynamic systems. So they're very like-minded with us, and we believe that based on the data they have in some of these scalable systems and the data we have, there'll be quite a convergence of our platforms, which really makes it much more efficient as we drive their programs, now our programs, to the clinic.
Got it. Maybe you touched on this a little bit, but how does the current financing climate impact your CMC investment decisions?
Right. So we were really fortunate because we built our GMP facility in 2021 into 22. And if you remember, our IPO was in June of 2021. I think you certainly remember the financing climate in 2021. So money was there for us, and we were able to build our own. I think it's really tough for companies today to put that kind of money into a facility. And to run it, if you do it right, it's not crazy expensive, but to build it is not cheap. And then to staff it is not easy. I always say when people ask me why build versus CMO, I'm like, well, there's three things. You need to have time to build, you need to have money to build, you need to have expertise to build and run. So time was on our side because we had the still have the FCDI relationship, which allowed us to make our first product with them. So we weren't in a rush to build this facility. But money was the big one. So back to the financing climate, since we had the money then and we built it, now we're well positioned. So we have a lot more flexibility to make choices that maybe aren't as dependent upon the difficult financing climate when it comes to manufacturing choices.
That makes sense. And just from a regulatory standpoint, are there any additional analyses that regulators have been wanting to see for iPS-derived products, or just in general, any kind of feedback you've gotten?
Yeah, definitely. So I think the fact that our cells are derived from iPSCs, which went through a series of reprogramming steps to get them, and the fact that we do multiple edits of our cells, even if we did a single edit, and now the fact that we have a single cell clone, which can be very well characterized. Yeah, there's been a big ask of us by the regulators to really understand that we have on-target integration and no off-target, which our processes are designed to do that. We have direct editing of site-specific editing, so we are doing it in a very precise way. But we have to prove it, so we have to prove that we have no off-target. But then when it comes to the edits and the differentiation and the fact that you're starting with an iPSC, there's always the risk of variance. So we need to understand what variants could be generated or are being generated, and then do risk assessments on those. So we use a whole host of genomic tools to understand what variants might be there. And then we have to then analyze the risks of these and then decide whether the product is still safe. And we've been fortunate so far. Obviously, we have a IND that's gone through the FDA. So obviously we feel that we and the FDA both feel that our risk assessment proved that our product is safe enough to try on patients, and so far so good. I can't disclose any clinical data other than what we disclosed back in December. We'll have more data cut soon, but we feel it's a very safe product. But we had to do quite a bit of genomic testing, both to prove the integrity and the stability of our edits and of our cells and our process.
That makes sense. All right, I think we're just about at the end of our session. So again, I'd like to thank you again, Greg, and we'll see you back in a bit for our cell therapy panel.
Thanks so much for the time and the opportunity. Looking forward to talking more later.
Great.
Thank you.